Good afternoon, dear students, ladies and gentlemen. Welcome to Histology Department of Medical Academy, named after Georgievsky. I am Associated Professor of Histology Department, Lugin Igor Anatolievich, and I will carry a lecture at the beginning of this semester. Today our topic is Introduction to Histology and Methods. At the first slide you have seen the symbol of our medical academy. This is of course the symbol of medical academy, which is represented like mythical animals, griffons, and the special symbol of medicine and flag of Crimea Republic in Russia Federation. And another, we have seen the symbol of Crimea Federal University also. This is mythological animal. Griffons is a symbol of Crimea. And at the center of this picture represented the symbol of our International Medical Faculty. We will waiting for you in our practical classes and of course in the Dean office. Please, let's start now. Histology is the science which studied the structure and functions of cells and tissues in the system of human body. You have seen here the classical microscope of beginning of 20th century, and we also have seen the cryostat, the special device for freezing section of the tissues, and of course the modern technique of fluorescent microscopy and confocal microscopy. So first of all, let's talking about history of histology. The first period before microscopic was started 2000 years before, 2000 years ago, sorry. And we have seen the Aristotle and other ancient philosopher who was to founded the methods of scientific research. But of course, that doesn't present any modern medical investigations, because the first microscopes was started to created around the middle of 17th century, and second microscopic periods continued around 300 years. The Marie-Francisque Xavier Bichat, one of the founder of histology and pathomorphology, have to name this as science and to give in the two special classification, one of the first classification in the history of histology, classification of tissues. The third modern period started at the 20th century and continued now. In this time we have seen the special methods like electron microscopy, immunohistochemistry, cytophotometry, autoradiography, confocal microscopy, and we have seen the one of the founder of embryological school in Crimea, Boris Khvatov. This is his photo. So what about subdivision of histology? Histology very close with join with some another biological science like cytology. Science about the cells and functions of cells in human body. Embryology, science about the development of embryo and fetal development, prenatal time. Also we have seen the special science which is present special between the histology and physiology. That's named histophysiology. This is science to investigate physiological process in tissues during life of living organism, and of course histochemistry, the science about methods of identification of chemical and biochemical substances inside the cells and tissues during the life process in living organisms. Also, for teaching histology, for representation of histology for students, we have classified in Russia histology like subdivision of general histology. This is a division of histology which investigates common tissues and special tissues. And the special histology, which investigates the system of organs, functional system and organic system inside the human body, with a combination of interactions between tissues, differentiation of tissues, development of tissues, histogenesis during the ontogenetic transformation of animal and human organism. So let's continue our talking about the microscopic period. A microscopic period was started in 1665. Then Robert Hooke, English scientist, observed a thin slice of cork under the microscope, which he was made it himself, and he saw that the composite of the cork tissue into the holes enclosed by wall. He is named the cells. That's book of Robert Hooke named Micrography, and contain also investigations of many living objects like insects, some other plants, but of course we have seen the classical Robert Hooke microscope consists from tubus and ocular lens and objective lens. That's composition of the microscope, like special device for investigations of the object of microworld, is named compound microscope. Compound because consists from more than two system of lens. And of course we have seen another famous person. This is Antonie van Leeuwenhoek. In 1677, Leeuwenhoek was been a Dutch maker of microscopes who made pioneering discoveries. His microscope very simple because it contained only one metallic plate and one lens. But this lens magnified the images in 300 times, like a modern laboratory microscopes. It was being amazing. And during his investigations, he's discovered the protozoa, red blood cells, capillary system, and especially was interesting that first spermatozoa of humans was being described by Leeuwenhoek works. Next moment, this is beginning of embryological investigation with microscopic technique. In 1681, famous Italian biologist and physician Marcella Malpighi to provide the series of investigations on the chicken embryo, which to result demonstrating the somites, nerve groove, optic vesicle, neural folds, and of course also describe red corpuscles like red blood cells and capillaries, movement of blood inside the capillaries in rabbit ear. So we have to talk about this fundamental scientists because their work to provide the formation of methods of histological and microscopic anatomy investigations. Origin of histology was started in 801. Then the French military doctor Marie-Francisque Xavier Bichat, he was the first investigator to discern the textural differences in the various parts of the body and to use the term tissue, as you have heard this is French word. Before, Bichat in English language not use the word tissue. Bichat classified 21 types of tissues in the human body, and his work became the basis of modern histology and pathological anatomy. His slogan, "Let's say life is an ensemble of functions that resist a death," will now transform into the slogan that a tissue is an ensemble of cells with the common functions. So you have to know that fundamental theory about cells, all living organisms, is named cell theory. It was being founded in 1839, 1838 time, and of course the author of this is fundamental theory was being Matthias Schleiden and Theodor Schwann, both German scientists, to have to postulate the main fundamental phrases. That's cells is a basic unit of all life. The second, structure of cells is similar for different organisms. And Rudolf Virchow, German pathologist and founder of cellular pathology, added these main principles like cell reproduction take place on the base of the principle omnis cellula et cellula, by division of cells, and continuous development of a cell theory to edit the next fundamental phrase, that's integral system of multicellular organism consists from tissues and organs which is regulated by intercellular, humoral and nervous mechanism of functional controlling. Well, thus you understood that cell theory is a base not only for cytology but also for histology and microscopic anatomy. We're talking about our medical academy. Medical academy, it's a part of the Tavrichesky University, which is named Crimea Federal University. The Tavrichesky University was founded in 1918, and the first department which was being organized in the medical academy, in that time in university, was the anatomy department, and the second is histology department. And the first director, Roman Gelvik, invite another famous professor, Alexander Gurevich. Alexander Gurevich was a famous Moscow professor who was to discover the mitotic division in the sea starfish and amphibians, and result of his investigations postulated the theory of mitogenic race, which is a base for theory of biological field. By the way, what does it mean, this mitogenic race? Then all living cells divide during mitotic division. They release specific ultraviolet rays. This is rays of life, as like Gurevich named, but later he was named mitogenic rays. That's fundamental work was being written in our university here in Simferopol, in Crimea, and after that printed in Moscow, and will stay one of the interesting theory of biology and cytology. Next, we're talking about our Crimean Scientific Embryological School. This year we celebrate 81 years of this scientific school. We have seen here, of course, one of the early stages of human development, the two pronuclei of zygote before the fusing is visible here, was made in our department. And also we have seen here the fetus in amniotic sac, and we have seen here the chorion. So long story of embryological investigation represented more than 10 famous scientists, like founder of the embryological school, Professor Khvatov, Grigory Petrov, Professor Shapovalov, Professor Trotsenko, Professor Georgievskaya, Korolev, Brusilovsky, and many other, and also Professor Borsukov. So these all people to provide the long embryological investigations and to describe all stages of human development, from the early stages, 17 days of development, embryo is named Crimea, till the methods of in vitro fertilization technique. That's methods, in vitro fertilization technique, to propose famous Professor Boris Khvatov. In 1942, he describes some interesting methods and cooperates with the American and English scientists to their investigations. That's very important that our department was been the first in the Soviet Union where is to investigate the fertilization technique. And his student, Grigory Petrov, in 1955 realized this methods of fertilization. We have seen here the device, special compound device, which contain embryonic chamber, also special gas, special elements of watering and gas exchange, and also we have seen microscope and the special camera. Result these activities, this is work, we have the unical photo of a human of early stages of development. That's after injection of spermatozoa inside the ovocyte, the spermatozoa nuclei will transform into the male pronuclei, and more light is female pronuclei. This is moment before the full completing of fertilization, fusing of pronucleus. And Grigory Petrov was posted the articles with Professor Khvatov, and these articles was being printed in a scientific journal and represented in Italy, where many newspapers to write this knowledge about the methods, technique of in vitro fertilization. Effect international students, that students from another country, foreign students, arrive to Crimea in 1961 after this news about the unical methods which is present in Crimea Medical University. Also, we have modern our investigation. And now let me introduce to you our professor of histology and embryology, Elena Yurievna Shapovalova. She's a doctor of medicine and science doctor, and she's a main scientific investigation is in vitro technique for fibroblast proliferation in skin, and embryonic investigation for toxical effect in mice and rats. Also, let me introduce to you the head of education, Associated Professor Svetlana Kharchenko and Associated Professor Inna Demyanenko. That teachers will carry on the practical classes in your group and also have to maybe be glad to see you in our practical auditory. And also we have the Associated Professor Galina Alexeyevna Yunsi and Associated Professor Tatiana Anatolievna Boyka. So you can join with teachers in Department of Histology or maybe in VK, our chart. Also, we have seen my photo and photo of assistant Svetlana Anatolievna Vasilenko. So we have a very big department. We also have seen here the Mrs. Marina Nikolaevna Zvereva. She is a laborant of our department, and we have unique symbol of our department, which to show the embryo inside cells. Okay, very important information for you. Please pay attention. How to find the materials for student in our site of Medical Academy, named after Georgievsky. You can find the main page. And after main page, you can see in the structure, take structure, click into the departments, click Department of Histology and Embryology and find materials for students. In this case, you have seen Module 1, Module 2, Electron Microscopic Micrograph. Electron Microscopic Micrograph, this is tools of electron microscopic photos of organs and tissues, cells, which using for pass the exam. Also, we have seen in Module 1 some topic like histological methods, plasma membrane, nucleus, mitosis, comparative embryology, and some other. And like example, we have seen the short prescription for each topic. Example, light microscopy, total magnification, fixation, embedding. So that's our lecture today described more effectively, but this is short prescription which can to help you, especially if you miss practical classes. Okay, next, this is special books and practical book which to represent it for students study. Example, main book of our department is General Histology and Nerve System. This is book, author is Elena Yurina Shapovalova, and also we have seen here the practical book for everyday home task. In this practical book you have to answer on some questions, to fill the some tables and designate electron microgram and schematic diagrams. Every practical classes current work. Well, thank you for that. And now we have to talking about introduction to histology and methods. Started from definition. What does it mean? Micro techniques. Micro techniques are different methods used for preparing histological section suitable for microscopic examination. Micro techniques include preparing thin section from the piece of a soft tissue. After that, staining sections. First of all, we have different methodics. Like example, the paraffin technique. It's the most commonly used. Used not only in our department, used in all over the world, like it's a classical routine technique, paraffin technique. Second is a freezing technique is the most rapid, but has some disadvantages. Some advantages for biochemical investigation, some disadvantages with the the quality of these investigations specific. We have to discuss a little later. Next, the Saladin technique is the most perfect, but modern methods ordinarily use paraffin technique like unical. Next, what about paraffin technique? Paraffin technique is used in preparing very thin sections, 5 to 8 microns thick. 5 to 8 microns thick. Ordinarily, now we use the experimental animal, like example rats. And we also have seen here the steps of paraffin techniques. Tissue sampling, next, fixation, step number 3, dehydration, step number 4, clearing by xylol, step number 5, impregnation in several changes of warmth of paraffin, melting 50 Celsius degree, and embedding in the molten hard paraffin, melting 55 Celsius degree. Next is a sectioning, and number 8 is mounting the paraffin section. Let's talking about each of them. First of all, tissue sampling. A small tissue sample is obtained by biopsy, diagnostic sampling, or post-mortem. In this case is named autopsy. So biopsy we use for investigation of living body, and autopsy for dead body. And suitable, it should be very small. Like we have to see here, the one of elements often for tissue investigations. If we use animal experiment, is named vivisection, to cutting the part of the animal organs and maybe any organs. Also, we use the investigation of smear. But what about biopsy? Needle biopsy we use for investigation of brain, eye, thyroid, lymph nodes, breast, lung, liver, kidney, bone marrow, testes, skeletal muscle. And endoscopic biopsy, it's another method without a needle. And in this case, we use a special endoscopic device, which to take in respiratory tract, in alimentary tract, urinary tract. And after that, also a special transvascular biopsy for heart and liver. And after that, we have to put this tissue sample into the special fixating substance. Like example, this is fixation is done by putting the tissue immediately in a fixative 4% formaldehyde in saline, which is now is using 10% formalin. And aims of fixation: preventing post-mortem degeneration by destruction of lysosomal enzymes, preventing putrefaction by killing bacteria, hardening the tissue by coagulating proteins so it becomes easy to cut into the thin section, and chancing staining of tissue. And how we do this is fixation. First of all, this is perfusion, when we use the special injection of injection of formalin inside the organ. Like example, perfusion of lung, maybe perfusion of heart. It's possible, but ordinary for dominate part of the organs, we use immersion. In this case, the tissue sampling put it inside the formalin bath. And next, dehydration to use for remove the water from tissue after the watering. By the way, why we need to do it? Because this is formalin. Also, we know about acetone and maybe alcohols in some methods, and liqua carnua, liqua buena, it's a more difficult mixture for fixation, is a ordinary poisoning, and we need to remove this poison from the tissue by the water, and watering continued from 1-2 hours till maybe sometimes 24 hours. It depends on the diameter of the organs. Next, dehydration. Replace water in tissue by alcohol. It's done by passing the fixed tissue in ascending grades of alcohol, 50 degree, 70, 90, 100, ascending, as gradual withdrawn the water by alcohol to minimize shrinkage of tissue, minimize the shrinkage of tissue. Like example, dehydration gradual is ascending grades of alcohol bath, 50, 70, 90, 100. And after that, we put the piece of tissue into the xylol. This procedure is named clearing. Clearing by xylol. The aim of this clearing, replace alcohol by xylol, as xylol is paraffin solvent. That's using for paraffin techniques. Make the tissue translucent. And you have seen that this red is scarred because after the xylol bath we can impregnate into the soft paraffin with a melting soft paraffin with a temperature 50 degrees of Celsius. Aims to replace xylol with paraffin, which penetrates the tissue. Embedding the molten hard paraffin after that will be more hard and higher temperature, 55 degrees. Also, embedding is done by putting the tissue into the cast containing the hard paraffin. Paraffin hardness as a cool forming the paraffin block, which can be cut into the thin section. We have seen how is visible. This is paraffin block. This is a paraffin, hard paraffin area. Inside we have seen the tissue sample. And after they're using the microtome, special device for cut, we have to start next process, which is named sectioning. Okay, so before that, again repeat the degradation in histology. You have this threads, which is like journey to the roller coaster pathway, because we have ascending and descending area of alcohol baths, maybe series of alcohol baths. Look, this is ascending fixation, alcohol 50, 70 gradual degradation, 90, 100 absolute. After that, clearing xylol over the 55, soft paraffin impregnation, embedding in hard paraffin, and we ready for sectioning. And after the sectioning, again xylol, absolute alcohol, 90 alcohol, 70 alcohol, distillate water. After that, we added hematoxylin, like special histological stain, basic stain, which alkaline nature. And the next is running tape watering, and next is the eosin. Eosin, it's acidic stain. And we have to colorate the histological slide. But back for sectioning technique. A A sectioning technique was being found by the famous European scientist Jan Purkinje. And Purkinje, you may be known about the Purkinje cells in a histological book, very famous. And we have seen that this microtome was being constructed by Purkinje more than 200 years ago. And microtome cutting into the thin serial section, the diameter of this section is a 5-10 micrometers thick. We have seen here the drive wheel, block holder, paraffin block with the tissue also visible, and steel knife. So that knife is very tiny and very sharp, can be cut very fast. Effect of it, we have mounting the paraffin section. Mounting is done by putting the paraffin section on a slide, which becomes ready for staining. Look, this is a paraffin ribbon with a tissue. Look, this is ribbon to put into the water bath, and this is water bath is not very cold, is not very hot, it's like a middle around 30 degrees, and in this case we have to catch each slide by special glasses, preparing glasses, and after that to demonstrate the put this is elements in the center of the glass. And that process is named mounting, because this is tissue is need to stick for the glass by a special glue. This special glue is not very difficult. We use the protein of chicken egg and added some timine for help prevent the bacterial invasions. So next, the freezing technique. In freezing technique, fresh tissue is rapidly frozen by liquid nitrogen or carbon dioxide snow. Sectioning is done in a cryostat, which keeps the knife or the microtome and the tissue to sub-zero temperature. Advantages of the freezing technique. This is a quick and rapid can be used by pathologists for quick diagnosis of urgent conditions. Can be used the histochemistry as no chemical solvent and used and the chemistry of the cells preserved. Disadvantages of freezing technique. Frozen section are thick and difficult to be cut and stained. Cryostat. Cryostat we have seen any minute ago. So here is represented the classical routine methods for identification of type of tumors. That's like a Mohs technique. And we have seen the tumorigenic area under the eyes. We have to remove with a lancet knife, and also we have to frozen section with the cryostat. After that, look under the microscope and to using the special histochemical methods for identified is this residual tumor or free tumor, and to decide in the oculistic surgery for future treatment of the person. Okay, let's now talking about light microscope. Components of light microscope may be known, but we just to remember some of them. The frame, the form that the base, arm, stage and tube. The magnified system from the condenser lens, the objective lens with magnification power of, for example, 10, 40, 100, the ocular lens or eyepiece with magnification power, for example, 5, 10, 15. and illuminating system is the source of light or mirror or sub-stage electric lamp. The famous photo of Leica microscope is present in many books, and we have just to, I only remind to you that this is a mechanical portion, arm, also a coarse focus adjustment knob, fine focus adjustment knob, which use the focusing of focus length in the microscope, stage adjustment knob, and also we have seen here the illuminating system, light intensity control, and also we have seen here mechanical stage. Also we have seen here objective lens, noise piece and body. Also we have seen the ocular lens. That microscope is compound and by is also with double objectives, double oculars, biocular microscope. But you also need to know that this schematic diagram to show the migration of light inside the microscope, and we have seen the presence of some prism. Prism is present in the body of microscope, and also prism is present in some condenser lens. Also, this is prismatic elements for using prevent the aberration, achromatic aberration. Also, the image in microscope is a virtual image. It's rotate opposites your investigation area. And if you need to watch the any part of the microscopic slides, you should remember that microscope it's virtually rotate this image for your eyes, because if you want to watch some area, you need to rotate the microscopic slides. Microscopic slides need to rotate opposite for investigation special areas of slides. Well, so what about magnification power of light microscope? The magnification power of light microscope, the power of objective lens increase in power of eyepiece. For example, when the power of objective lens is 40 and the power of eyepiece is 10, so the magnification power of light microscopes 410 increased to 40 is 400 times. Well, and in a microscopic technique we use a special measurement. This is measurement, this name, not millimeters and centimeters. We use the ordinary micrometer or micron. This is 10 minus 6 time, and also we use the nanometer. A nanometer is 10 in minus 9, and also angstrom, 10 in minus 11. So this is a special measurements for microworld. Look at this schematic diagram. We have seen that naked eye have shown for us the centimeters, millimeters, but the level of 10 micrometer plant cells, animal cells, invisible for naked eyes. For that we use a light microscope. But if we continued the investigation, and if we need to know how they are organized, not a finger or epidermis, we need to use another type of device, electron microscope. That electron microscope can show for us not only cells of animal or cell or plant, but also bacteria and viruses and ribosomes. In this case, it's shown the magnification and resolution in 100 nanometer, 10 nanometer, and more than 1 nanometer in angstrom. In this case, we're talking about the molecular and atomic level. Well, so what is resolution? Resolution power. The resolution's the degree of separation that can be seen between a just point to allow us to see these points as separate points. Power of human eye is 0.2 millimeters, but the resolution power of light microscope is 0.2 microns, and the resolution power of electron microscope is 0.002 micrometers, and more than is sometimes 2 nanometers. Look, this is example. What does it mean? Separation between 2 points. If you have seen it here, is very good visible, but smaller diameter you're not seen. But if we have magnified, you're very good seen again. So because that, the famous German physics scientist Ernst Abbe, to define the resolution, microscope resolution, like ability of a lens to separate or distinguish small objects that are closer together. Wavelength of light use is a major factor in resolution. Look at this one. This is a formula, mathematical formula. Resolution is a lambda is a wavelength to divide on the 2n sinus alpha. What is it? 2n. N, it's a numeral aperture. And the sinus alpha, this is the angle between objective lens, which is represented here like numeral aperture. Example, this is short focal length, more magnification, and short wavelength, great resolution. Example about the structure between objective lens and objects. This is the angle. This is angle can be changed as an effect of environment. That environment maybe is air, and maybe this is oil if we have to use special immersion objective lens, immersed objective lens. That's ordinary 90 and 100 magnified objective lens. This area is named working distance. Distance between the front of surface of lens and surface of cover glass of specimen. And how are we to use for our microscopic investigations? Look, this is structure of objective lens. This is achromatic and apochromatic. Special reduce the aberration, chromatic aberration inside microscopes. Modern objective lens can be classified only 3. This is the small magnification, 8 and 10 times magnified, middle magnification, 40 magnified, and 90 or 100 magnification. In this case is named large magnification objective lens. Magnification to also characterize numerical aperture. Numerical aperture, this is a diameter of lens inside objective, and we have seen that's like low power 10 magnified in 10, ordinary 0.25, magnified higher power 40-45, it's 0.55, 0.65. Oil immersion objective lens is ordinary 1.25, 1.4. Approximately focal length also represented here, working distance also represented here. So you can use this as special tables for your microscopic investigations. When you buy a classical modern trade famous like microscope like Leica, Carl Zeiss, Olympus, you can to find this is special scale of table in your microscope. Okay, that's a main moment of the microscopy. This is condenser lens. Condenser lens and type of objective lens. Because that we have classified light microscopy in too many types, like bright field microscopy. This is classical microscopy which you study in school, maybe in biological laboratory. Special division of this microscopy is named dark field microscopy, phase contrast microscope, fluorescence microscopes. All these microscopes are compound microscopes, an image formed by action of more than two lenses. Shortly describe these methods. The first is dark field microscope. Produce a bright image on the object against a dark background. Look at this one. Used to the observed living unstained preparation. Very famous now, because not all laboratory can buy the modern, very expensive histological stain and histochemical stains. But if you can to use dark field condenser, like dark field stop elements in the condenser, Abbe condenser is named, you can to use this method without special stain. And the fibroblast cells is very good, bright inside on the back black ground surface. Okay, the phase contrast microscope enhances the contrast between intracellular structure, having the slight differences in refractive index, index of refraction of the light, and of course, excellent way to observe a living cells also. Look, what is this? Is the result of diffraction rays are retarded the 1 and 4 wavelength after the passing through the object. So this is name is face plate and face ring. The face plate is represented inside the condenser lens, but the face ring is represented inside the objective lens. Effect of it, we have the unical instruments for the formation of like tridimensional image, maybe with a different contrast reaction, contrast result. It's also fibroblast. Look at this next methods, very interesting. It was being result of great foundation of Nomarski. Nomarski was a Polish scientist who emigrated to United States of America and got the Nobel Prize for construction of this specific condenser prism. Condenser prism of Normansky creates the image by detecting differences in refractive indexes and thickness of different parts of specimen. We have seen the fish egg division. This is early fish embryo in the stage of two blastomeres. Is very good visible. That's envelope of fertilization, saving, and we have seen the blastomeres, very good visible nuclei. So this method is named differential interference contrast microscope method. Well, so also not need special stain. And the next moment, this is the very modern technique, fluorescence microscope, exposes a specimen to ultraviolet, violet or blue light. Specimens usually stained with fluorochromes. That method is named fluorescent. but also we know luminescent. What is it, luminescent? Luminescent, then the ultraviolet rays to use for refraction of this outermost luminescence. Like example, we know that natural luminescence represented for nerve cells, for medulla of suprarenal gland, chromaffin tissue, and for sperm cells, as like in forensic medicine, when the detective who made the investigation, criminal investigation, can find the sperm cells elements if was present, like example in criminal forces in the victims of the forces, and we have to use these as elements like luminescent investigations. But in histology, luminescent not very often used. Ordinary use fluorescent. So fluorescence is also used the fluorescent light. This is named ultraviolet light. So with especially mercury vapor or lamp, mercury lamp. This is mercury lamp to have generate the ultraviolet rays, and this is rays to migrate on the area of the where is present dark field condenser lens, also like in previous investigations. And another area is this is a barrier filter. Effect of this is investigation. We have seen the bright image of the object resulting from the fluorescing light emitted by the specimen, and the different area of tissue, different area of cells characterize fluorescence, different images like different bright coloring, different coloring. Example, DNA of nuclei, it's a coloring by blue. This is a green color. This is a microtubules, cytoskeleton of fibroblast cells. And another, this is myofilaments. Myofilaments is actin filaments, is very good visible at the periphery. Ectoplasm is named in cells. And not in other methods can be identified except fluorescence microscopy. That's fluorescence microscopy very popular not only in classical microscopy, but also in fluorescent immunohistochemistry for identification of different type of proteins. Let's now talking about very modern techniques, like a famous is confocal microscope. This is classical photo of the modern confocal microscope, inverted confocal microscope. Look, that's in confocal microscope, that microscope is named scanning and use the laser rays. Laser rays source is like a special laser gun, can be provide the migration of some rays, laser rays into the object. But when laser rays is migrate, that's back and catching by the beam splitter. This is beam splitter reflected and divided them, and to direct this is rays into the special plate pinhole which to migrate into detector. This is detector joined with computer PC. Like another moment, this is a migration of laser source. This is laser source is migrated and like scanning the surface of specimen and to change the focal plane many times. Effect of it, the combination of different focal plane with computer analyzing, and the result is tridimensional, very coloring very beautiful image representation. Resolution, in this case, very high. We just can we have to see two types of microscope. This is conventional light microscope, biofilm, and also confocal scanning laser microscopes. Laser beam used to illuminate the spots on the specimen. Computer compiles images created from each point to generate a three-dimensional image with a higher contrast. In this case, you have seen in the left image field of view is not contrast, but in the laser confocal microscopy is more detected, is more contrast, as more resolution is more higher for the identification of different types of cells. It's very good. Now let me introduce to you classical staining technique. Classical staining technique maybe you find in many books about the slides. That's hematoxylin and eosin. Hematoxylin is represented at the left first picture at the upper corner. Hematoxylin is a basic stain. This is stain made from the Campeche tree. So this is a natural stain. Campeche tree to growth in the Middle Africa, and Campeche tree to have to use the cork of Campeche tree for to make the hematoxylin. Of course, the cork of Campeche tree is a brown color, but when it's put into the water, will be blue or purple color. And another variant, this is eosin. Eosin is chemical, artificial stain which was been synthesized in the chemical industries for acidic substance. But this is acidic substance, very good affinity for alkaline elements. Alkaline elements ordinary a lot in the cytoplasm, but the acidic elements a lot in the nuclei. Because that we have talking about classical redox reaction. Redox reaction you may be studying in chemistry, and this is reaction it's take place inside the cells after the staining techniques. Look at the left picture. This is combination of chemical reaction. Blue-purple nuclei and pinkish and red granules inside the cytoplasm. This is hematoxylin and eosin methods double staining detection. Double staining detection, it's necessary for any morphological investigations in surgery, in histochemistry, in pathomorphology, many use. Another special methods also present here, trichromacy. Trichromacy or trichromic stain by Masson. Trichromic stain by Masson we have seen here, very good identify different types of tissue. Blue color, this is connective tissue, loose irregular connective tissue. Purple color, this is epidermis, different types of epidermis, 5 layers. And the last, the red color, this is stratum corneum, cornified area of skin, cornified area of epidermis. So this is trichromasia or trichromic stain by Masson, can be identified different type of tissue in the structure of an organ. Another methods more interesting for semi-thin slides, like example for analyzing before electron microscopic technique. That's methods used in this picture for kidney glomerula. And this is kidney glomerula. We have seen the numerous capillaries networks, and we have seen the podocytes, and this is stain is a special name toluidine blue. Also, we have seen Van Gison stain. Also, very good detect different type of tissue. Look, red color, this is collagen fibers, and elastin fibers and muscle fibers is different coloring, red and pink, red and orange. And another in the center, this is epithelial lining in the prostate gland. Okay, thank you for this part. And now, what about the principle of staining with hematoxylin and eosine? Look at this. That's we just can describe this moment in your lecture notes. I want to tell you, my dear friends, that the histological lecture is very important for you, because you need to copy some materials in your lecture notes. This is lecture notes you can show to your teacher, you can show to head of department, and you have shown for the current practical classes, like consultation, like questions for discussion of future topics. Like example, before any topics, you need to read a lecture and compare the materials from lecture with materials from classical textbook. As effect of it, you can double repeated the materials. After lecture, you have analyzing your notes. Before practical classes, you repeat it again after the reading the text of atlas and text of textbook. What about acidic structure? Acidic structures, ordinary DNA, desoxyribonucleic acid and ribonucleic acid, you know from the biology course, and that and biochemistry you will study more, but that's like nucleic acid is need to identify with basophilic stain. That's joint affinity with hematoxylin. So hematoxylin alkaline in nature and very good join with DNA and RNA. Eosine is acidic in nature, but very good join with alkaline components of cytoplasm. How we need to remember? We need to remember that nuclei, it's ordinary basophilic, that's word which used for description of microscopic slides, and the cytoplasm is ordinary oxyphilic or acidophilic, which used for description of organelles and cytoplasm. Well, look at this one. Again, repeat. Acidophilic structure and basophilic structure. Nuclei is basophilic, cytoplasm acidophilic or oxyphilic. And again, we have to find in all our microscopic slides. And now I am represent to you the photo of pituitary gland. In pituitary gland we have find three types of cells. That's its name as same as the identification of these colors. Example, we not say I have seen pink cell. No, it's incorrect. I have seen acidophils or acidophilic cells. This is acidophilic cells, very good stained by eosine, and maybe it's acidophilic. Another cell is basophilic, and is named basophils. Then the cells which is not good stain is named chromophobes, because not good stain, chromophobes. And chrome is a color. Phobos, it's a scared because not good stain, chromophobes. Acidophils is good stain by acidic stain like eosin, and basophils very good stain with hematoxylin, as name basophils. Okay, in the epidermis of skin we also have same composition. Again, nuclei, it's a blue color, basophilic, and the cytoplasm is more red color, oxyphilic or acidophilic. In the muscle, also same. The nuclei is like a rod-like structure. That is a smooth muscle cells. And we have talking about the nuclei is basophilic, but the cytoplasm is oxyphilic. Okay, next, this is procedure of steps of staining in hematoxylin. It's very useful for methods of staining. And first of all, after the sectioning, when we have to get normal glass with histological slides on surface, we need dissolve paraffin section in xylol 2-3 minutes. Put the slide in the descending grades of alcohol 190, 70 and 50, 2 minutes each. Put the slide in distillate water for 1-2 minutes. Stain with hematoxylin for 3-5 minutes. Wash in running tap water for 5-10 minutes. Counter stain with eosin for 1-2 minutes. Wash in distillate water for 2 minutes. Dehydrate in ascending grades of alcohol. Again, opposite, right? 50, 70, 90 and 100 for 1-2 minutes each. Put in xylol for 2-3 minutes. It's very important, not more than 2-3 minutes, because the slides will crash. And put immediately in Canada balsam. What is it, Canada balsam? Canada balsam, it's a wax of the of a Canadian pin, Pinus Canadiae, and this is a plant product, which to very good covering the cover glass, and use like a glue for the histological slides. You can see like yellowish color under the glass. And this is a mounting of slides. I congratulate that you have been study this moment and will have a joy during the histology roller coaster pathway for the ascending, descending investigations for the slides. If you can visit our laboratory for experimental histology. And again, repeated like ascending, gradual degradation, dehydration, gradual dehydration, and next is clearing, and next is again descending dehydration, descending dehydration. And we have all do it for very high quality of the histological slides, because slides should be translucent and absolutely good stain with histological double stain, hematoxylin and eosin. Primary hematoxylin, after that eosin, not opposite. And this is the result of our investigation, and you have to remember another stain, like staining fat with Sudan 3, orange color, Azan stain nuclei red and collagen blue, also stain carbohydrates with glycogen with periodic acid shift for identification of glycogen, stained magenta, and staining with silver impregnation stains many structures like reticular fibers and also nerve cells appear black, and histochemical staining and immunohistochemical, which is we just started a little late. And look, this is histochemical stains example. That's hematoxylin and eosin, you know, iron hematoxylin for identification of some cells, especially muscle cells, Mallory-Azan stain, Masson stain, trichromacy, periodic acid shift, magenta color, silver impregnation, black color, Orcine for elastic fibers, azure, Sudan black, alkaline phosphatase. And like example, periodic acid reaction for identification of glycogen in the kidney. Look, this is the kidney glomerula, it's more magenta color, so accumulates the glycogen's element. Okay, the next is a Felgen method. Felgen methods for identification of DNA and the chromosome, it's a more purple, not like blue color cytoplasm. Immunohistochemistry, we have seen here the identification of microtubules in fluorescent microscopy of decidual cells in rabbit, and this is, we have identified the basal cells in prostatic gland duct. All another cells is blue and red, blue and pink, so it's hematoxylin, but this is brown color, it's basic cells, which is basal cells, which is identified with phosphatase, alkaline phosphatase. Okay, what does it mean, immunohistochemistry? Immunohistochemistry, first word immune, it means that B lymphocytes, like a specialized lymphatic tissue cells, to provide production of plasma cells, and plasma cells produce antibodies and produce maybe immunoglobulins. G immunoglobulins, same like example, and we have to find these methods for two variants. The first variant to production of monoclonal antibodies, which is very unique for special type of receptor, special type of antigens identified for every type of animal. Example, mouse antibody we use for identification of human's tissue, rabbit antibody for identification of rat's tissue, and we have to use a hybridoma. So this is hybrids, monoclonal antibodies can special colony of lymphatic tissues for production is used in vitro methods for cloning of the cells, and was made in a special laboratory. Another is polyclonal antibodies. It's ordinary use for several different species, but not unique, not can very often use for identification of some pathological process. For pathological process, very often use monoclonal methods. And let's example stages for this is immunohistological techniques. Also, we have two methods, the straight and non-straight methods. Antibody, we add it into the tissue with antigens. And indicator of this is reaction between antibody like immunoglobulins, which is joined with antigen like key and holes. keyholes and key. As a reflex of it, we have the coloring. Colorless substance can be identified to some area, and because the first step same, fixation, tissue sectioning, paraffin embedding, inactivation and blocking. After that, very important, antigen retrieval. Next is detection and chromogens counter stain and mounting media. Example, look, this cells is very good stain, and we have seen here how this visible in pathomorphological slides. Okay, sorry for time, we need to continue very faster, and next moment the mechanism of electron microscopic investigations. We have two types of electron microscope. Transmission electron microscope, TEM, visualize the internal structure of the cells. Scanning electron microscope, visualize the surface of cells, three-dimensional image on the screen as microvilli and cilia. Look at this picture. The main part of the microscope electron type is cathode anode. That's his name, electron gun. That's a construction was been made by Ernst Ruska. And Ruska was been German scientists who were the first to provide this method for higher voltage electrode, cathode and anode start higher temperature release the electron beam. And this is electron beam can be migrated through the series of the condenser lens, electric coil, objective lens, intermediate lens, projector lens, and all is collected inside the fluorescent screen or special camera which to make the photography. This is classical original microscope in the laboratory. We have seen that electron beam is in descent on the surface, but instead of using the transmission electron microscope. The source of illumination is beam of electrons emitted from the heated tungsten filaments, cathode. The beam of electrons are attracted to the anode. Set the electromagnetic lenses. The beam of electrons passes through the series electromagnetic lenses which serve the same functions as the glass lenses in light microscopy. We was talking about a minute ago, and the final image is received the view by the fluorescent screen. Look at this one, how is visible. This is classical transmissional electron microscope to show for us neutrophilic leukocyte, blood cells. We have the nuclei, it's segmented. We have is more dark area and more light area. This more dark area is named electron dark area. That's heterochromatin. Electron light area is named euchromatin, but originally is named electron lucent area. Look at this one. What's the differences between light microscopy and electron microscopy? Nature of the physical forces. Daylight or substage lamp, beam of electron, glass lenses, electromagnetic lenses, and resolution power 0.2 micrometers and 0.2 nanometers. And magnification also different. And look at this one as a scanning transmission electron microscopy. It's a secondary electron reflected beam. Elasticity, the elastically scattered electrons is detected because the scanner is moved with a specimen holder inside. This specimen holder also located in the vacuum chamber, but is moved. As a fact, we have tridimensional image of nuclei, mitochondria, plasma membrane, lysosomes. And look, Description of electron microphotos. We can use the term electron dense and electron lucent. Electron dense regions in the specimen scatter electrons and thus produce white area on the negative. Because the photograph printing reverse, black and white areas on the negative appear black on the print. Accordingly, the electron-dense region of the transmission electron micrograph appear black. So, if we have talking about heterochromatin, we name it electron-dense area. If we're talking about euchromatin, we name it electron-lucent area. Example, this is red blood cells inside the capillary, electron-dense. But this is a basement membrane in the glomerula. This is electron-lucent area, and especially electron-lucent plasma. Also, we have seen scanning micrography of this is glomerula of kidney, glomerula of kidney. This tridimensional image, more good for investigation, but less in resolution. And another preparation methods, shadowing. For investigations of insects and some organs, we use coating the specimen with a thin film of heavy metal. In this case, we have investigates tridimensional image fully and colorized with special techniques. The freeze etching. Freeze etching the specimen when fracture along lines of greatest weakness after the frozen technique with a liqua nitrogenum. As effect of it, we have seen the structure of tridimensional nuclei and tridimensional mitochondria. And the last method we study today is autoradiography. The autoradiography is a bioanalytical technique used to visualize the distinguish of radioactive labeling suitable with the isotope in the biological sample. It's the method by which radioactive material can be localized within the particular tissue, cells, organelles, or even biomolecules. It's a very sensitive technique because to show for us the level of biological process, like example, biosynthetical process. We can identify level of biosynthesis, the process of biosynthesis of protein, process of activities of genes, and we have to development of autoradiography as biological techniques, but very fast developed after the Second Great War, and we have the interesting of labeling with radioactive isotopes. Ordinarily, we use tritium. Tritium is hydrogenium-3, mass 3. And this is tritium can be used in the special organic substances like timidine and uridine. It's analogous of timine and analogous of uracil in DNA molecule. DNA and RNA molecule. Like in DNA, for DNA we use timidine, for RNA we use uridine. So as a result, we have seen the tracks. This is spots, black color spots in different magnification. This is area where it accumulates the timidine. By the way, no. So this is a granule. So this is maybe uridine, accumulates uridine. And it's shown the level of biosynthesis of protein. So autoradiography based upon the ability to radioactive substances to expose the photographic film by ionizing it. In this technique, radioactive substances put a direct contact with a thick layer of photographic emulsion and need exposition around some minutes. Effect of it, we have to identification of silver molecule on the surface. Silver halide crystals are exposed to the radiation which chemically converts silver halide into the metallic silver, giving the dark color band. And effect of we have like electrophoresis and digital scanning. Example methods present here. This is the steps of activation and identification of the special silver molecules. Like the results is reduction of argentum silver ions to argentum atom, leaving the dark color bands, and the slides when washed away by fixers to get the insoluble argentum ions only. The outer radiogram can further the viewing the observed under the microscope. So, this is combination of radioactive method and methods of photography and for identification of biosynthesis of protein and gene realization. So application, we have to identify the level of DNA, properties of DNA and RNA also. Thank you very much for your attention. I hope you will necessarily visit it next lecture. We will glad to see you. If you need some questions, please ask in the special chat of VK, which is named Department of Histology and Embryology. I will be glad represent to you that materials. Thank you very much again.